This research was conducted for my undergraduate honors thesis and studies the prevalence of Streptococcus pyogenes, Staphylococcus aureus, and Methicillin-resistant Staphylococcus aureus in high-traffic areas. My name is Carlie Fusco, and I am a part of the Department of Biology at Francis Marion University. This thesis was mentored by Dr. Jennifer Lyles of the Department of Biology at Francis Marion University, Dr. Lorianne Turner of the Department of Biology at Francis Marion University, and Dr. Jessica McCutcheon of the Department of Chemistry at Francis Marion University. Many bacterial species are transmitted from humans to fomites, or inanimate objects, every day. Consequently, individuals may become infected with these bacteria simply by coming into contact with common surfaces. Streptococcus pyogenes, which commonly causes a condition known as strep throat, is a bacterium which causes the throat to swell. Streptococcus pyogenes is most commonly spread through direct person-to-person contact by saliva or nasal secretions from an infected person. Staphylococcus aureus is an opportunistic bacterium that many people carry on their skin and about 30% of people carry in their nose. Opportunistic means that the bacteria is usually harmless unless it enters the bloodstream. Many skin infections can be caused by Staphylococcus aureus, including boils, impetigo, and cellulitis. Staphylococcus aureus can be transmitted from person to person and can survive on objects like pillowcases and towels. Methicillin-resistant Staphylococcus aureus, more commonly known as MRSA, is a bacterium that is resistant to several antibiotics. MRSA is a strain of Staphylococcus aureus and most commonly affects the skin. MRSA can be spread through contact with a contaminated wound or by sharing personal items like razors and towels that have touched the infected skin. 59 samples were collected from various surfaces in high-traffic areas of a healthcare clinic, a childcare facility, 2 gas stations, and 2 laundromats. Some of these surfaces included door handles, waiting room chairs, playground equipment, gas pump keypads, and washing machine handles. Sample collection was performed using sterile swabs pre-moistened in sterile phosphate-buffered saline, or PBS. Samples were enriched via incubation in Tryptic Soy Broth, TSB, at 37 degrees Celsius for 24 hours. Trypticase Soy Agar, TSA, was then inoculated with the enriched samples using the swab method and incubated. TSA plates were then observed. Plates with significant growth were chosen to be replica plated onto Mannitol Salt Agar, MSA, and TSA supplemented with 5% sheep's blood. MSA is a medium used for the isolation and identification of Staphylococcus aureus and turns yellow in the presence of Staphylococcus aureus. TSA supplemented with 5% sheep's blood is a medium used for the isolation of Streptococcal pyogenes. Positive growth of Streptococcal pyogenes presents on 5% sheep blood agar as beta hemolysis of the red blood cells. Beta hemolysis causes growth to appear yellow in color. Growth that indicated the presence of Staphylococcus aureus on MSA plates was then transferred using a sterile wire loop to MRSA Select II plates. MRSA Select II medium isolates methicillin-resistant Staphylococcus aureus by showing purple growth and inhibiting any other growth. On TSA plates, significant growth was observed from 52 of 59 samples after incubation. Of the 52 TSA samples replicated for growth on MSA plates, only 37 showed evident growth of Staphylococcus aureus. 24 of 37 MSA samples that were transferred on to MRSA Select II plates exhibited growth indicating evidence of methicillin-resistant Staphylococcus aureus. On blood agar plates, no evidence of beta hemolysis consistent with Streptococcus pyogenes was observed. However, evidence of alpha hemolysis was observed from 51 of the 52 samples after incubation. The results from this study suggest that Staphylococcus aureus and methicillin-resistant Staphylococcus aureus are prevalent on fomites in high-traffic areas of healthcare clinics, childcare facilities, gas stations, and laundromats, while Streptococcus pyogenes does not appear to be common on these surfaces. All sample collection was done during the midst of the SARS-CoV-2 pandemic. Assuming that masks were worn correctly and consistently, individuals infected with Streptococcus pyogenes were not spreading the bacterium as they would have without the mask. The likely transmission of Streptococcus pyogenes through saliva and nasal secretions via coughs and sneezes was inhibited or lessened by the wearing of masks. Additional studies, including molecular analysis, are required to determine the exact identity of the bacterial species and strains present. Here is a graph that shows the total number of samples that exhibited growth of each species. As we see here, there were 37 of the 59 plates that showed evidence of Staphylococcus aureus, and there were 24 samples that showed growth of Methicillin-resistant Staphylococcus aureus. Unfortunately, none of the samples that were collected showed evidence of Streptococcus pyogenes. Here we have sample 48, which was taken from a washing machine door handle. These photos exhibit the growth observed from sample 48 on TSA, MSA, MRSA Select II, and blood agar. Significant growth can be observed in TSA, and the yellow agar color change consistent with Staphylococcus aureus is present on MSA. Purple colored growth consistent with MRSA is visible on the MRSA Select II agar. The blood agar shows alpha hemolysis, not the beta hemolysis consistent with Streptococcus pyogenes. Beta hemolysis would present as yellow-colored growth compared to the green/brown appearing growth seen with this sample. Thank you for listening to my presentation, and for more information, you can contact me, Carlie Fusco, at cfusco5728@g.fmarion.edu.